Assalamualaikum warahmatullahi wabarakatuh. My name is Nurul AIDA bint RASHID. So today, the title of my study is about heavy metal accumulation and phytoremediation potentiality in mangrove plant. Heavy metal pollution in a mangrove is a significant problem that requires severe and urgent action from all concerned authorities worldwide. Heavy metal occur in the ecosystem from natural processes or anthropogenic activity. They can be released into the environment during weathering, which result in soil formation in a natural ecosystem since they are found in ultramafic rock and all mineral. Source of anthropogenic such as mining activity and urbanization became the main responsible for heavy metal pollution. As a result of their inability to be broken down, heavy metal gradually accumulate in mangrove sediment where they might endanger ecosystem. Other than that, heavy metal are also being transported by river that end up in diverse marine ecosystem, and mangrove is one of them. Mangrove can be classified as the most prolific ecosystem on the globe as it provides variety of ecosystem service such as erosion control, animal food, stabilization of coastal landform. Mangrove plant can be categorized as green barrier due to their extraordinary capacity to decrease the speed of heavy metal to other area. It can absorb the heavy metal from the sediment, store them in root, stem and leaf. A method known as phytoremediation uses the plant to absorb and eliminate toxic elements from environment to reduce the bioavailability of those contaminated in sediment. To absorb heavy metal and control their bioavailability, plants stretch their root system into soil matrix and create rhizosphere ecosystem, reclaiming the polluted soil and stabilizing soil fertility. Using plant as phytoremediation can be a promising way to revegetated polluted soil. Problem statement. Heavy metal accumulation in mangrove plant is a developing environmental concern due to the possible negative impact on ecosystem and human health. Various anthropogenic activities including industrial discharge, agriculture runoff, and urban expansion can cause heavy metal in mangrove environment. This heavy metal can enter the mangrove environment via sediment deposition and water uptake by mangrove plant where they can accumulate in plant tissue. Heavy metal deposition in mangrove plant can alter physiological processes, harm growth and development, and ultimately impact mangrove ecosystem general health and productivity. It also can infiltrate the food chain by consuming product derived from mangrove, posing a threat to human health. Using plant to remove or mitigate environmental contaminant known as phytoremediation has emerged a promising approach for dealing with heavy metal contamination in mangrove environment. Mangrove plants are suitable for phytoremediation due to their unique characteristics including high biomass production, extensive root system, and tolerance to severe environment. However, further research is required to determine the efficacy and potential of mangrove plant in phytoremediation of heavy metal. Therefore, this research seeks to determine the heavy metal accumulation in mangrove plant and their phytoremediation potential. Objective. The main aim of this study is to determine the concentration of heavy metal in Tanjung Karang mangrove plant. The specific objective of this study include to determine the concentration of heavy metal in mangrove plant by using ICP-OES. Second, to compare the concentration of heavy metal in between root, stem, and leaf from three different species of mangrove plant, which are Sonorachus, Celeris, Nipa palm, and Avicenna alba. Third, to evaluate the phytoremediation potentiality of mangrove plant by calculating the bioconcentration factor and translocation factor. Okay, next we move to literature review. There are a few studies that specify on determine the concentration of heavy metal in mangrove plant. As we can see from the table, the highest concentration of zinc in root and leaf was determined in landfill site by Amlazak in 2017. In Matang mangrove forest in Malaysia, Cu concentration in roots and leaf are relatively low at 0.006 and 0.005 mg per kg respectively. CD and Pb concentration were reported only in the study by Suraya in 2019 in Belanakan Pond Subang, Indonesia. This method shows variability in concentration across different plant parts, with Pb concentration being generally higher than CD. Overall, the comparison shows significant variation in heavy metal concentration in different plant parts across various study areas. Factors such as environmental conditions, contamination sources, and plant species influence this variation. Next, this table represents BCF and TF from various studies. BCF represents the ability of plant to accumulate heavy metal from soil into their root and leaf. BCF greater than 1 indicate that the plant has higher concentration of heavy metal in its root and leaf compared to surrounding soil. A study by Hussain in 2022 it showed that the BCF value for Fe, Cu, zinc and Mn were less than 1, suggesting that these mangrove plants do not strongly accumulate this heavy metal in their root and leaf. Study by Hunt in 2020. The mangrove plant has ability to accumulate Mn, zinc, Cu and Fe because the BCF value were mostly above 1. Next, TF greater than 1 indicate that the plant efficiently move heavy metal from roots to other part. As we can see, mostly the study conducted have TF value greater than 1. Next, we move on to the methodology. For sample collection, samples were collected from 3 different species, which are Avicennia alba, Sonoretia cacellaris, and Nipah palm. Leaves were chosen from healthy-looking trees with 4 to 6 cm long sample. Root and stem were collected from the same healthy tree. Sediment samples were taken from 0 to 10 cm deep. For sample preparation, root, leaf, stem, and sediment were dried in oven at 105 degrees Celsius until they reach a constant weight. The dried sample were crushed and passed through a 0.5 mm diameter sieve. Sediment then pulverized into a fine powder and was used to form pellet for analysis. For sample digestion, 0.5 gram of each dried and sieve sample was placed in separate vessels. Nitric acid and hydrogen peroxide were added to each vessel to create a solution. After digestion, the solution were filtered and diluted to 50 ml. Sample measurement. The concentration of element in the plant sample was analyzed using ICP-OES instrument for sediment sample, and EDSRF instrument was used for analysis. Next, for result of sediment, the order of decreasing concentration among the studied heavy metal was Mn, zinc, plumbum. Plant and sediment recorded the higher concentration of Mn and zinc compared to plant and sediment. The high concentration of heavy metal in study area may be attributed to anthropogenic sources such as aquaculture operation and sewage effluent. Comparison with earlier study revealed that the concentration of Mn and zinc in this study were higher than in some other location, indicating possible anthropogenic influence. The concentration of Pb in the sediment was higher in the current study compared to previous study, and the maximum observed concentration was 147.64 mg per kg, exceed the average shale value. The presence of higher concentration of heavy metal, particularly Mn, zinc and plumbum, suggests the possibility of lithogenic origin. Heavy metal can enter the ecosystem from human activity, contributing to long-term pollution. CD in sediment were higher than average shale value in other study. The concentration of Cr was only recorded in one location and varied from other study. Overall, the findings suggest that some of the detected heavy metal concentration in the sediment from different mangroves worldwide were higher than the average shale level. Next, the control root show higher zinc concentration compared to Nipah palm root, which can be attributed to inherent fluctuation in natural ecosystem where the control sample may coincide with area of higher natural zinc concentration. Avicennia alba root recorded the highest concentration for Pb compared to control plant. For root, the average concentration in order Sonoretia cacellaris, Avicennia alba, control plant, Nipah palm. Next, for Mn, concentration in stem, Sonoretia cacellaris has the highest accumulation compared to other plant in the present study. For stem, the order of average concentration, Sonoretia cacellaris, control plant, and Avicennia alba. For leaf, the order of average concentration, control plant, Sonoretia cacellaris, Avicennia alba, and Nipah palm. For Pb, Nipah palm has higher accumulation in their leaf, which much higher than the control leaf, 0.0037 ppm. It can be seen that the highest concentration of zinc was found in control sample. The concentration of zinc was high in control sample due to the plant may have higher uptake of zinc because they growing in an environment with more readily available zinc. Next, different mangrove species show varying concentration of zinc, Mn in their root system and leaf. The presence of Cr and Pb concentration vary among the studies. Zinc concentration in Avicennia alba, Soneratia cacellaris, and Nipah palm were in the range of 0.586 until 1.803 ppm, while Pukia indica show higher zinc concentration ranging from 12.825 until 20.320 ppm. Mn concentration in Avicennia alba range from 0.573 until 3.581 ppm. And overall, the data show that different mangrove species and location exhibit varying concentration of terrace metal, emphasizing the importance of understanding the distribution and accumulation patterns of this metal in coastal ecosystem. Next, the bioconcentration factor was used as indicator of species accumulation ability from sediment to different body parts, root and leaf of 3 mangrove species. The result shows that all the plant organ, root and leaf had BCF value less than 1 for all heavy metal. The highest BCF value was found for zinc in the roots of Avicennia alba, while the lowest BCF value was observed in Nipah palm. For Mn, the highest BCF value in root was found in Soneratia cacellaris and the lowest was in Nipah palm. For Pb, the highest BCF value was found in Avicennia alba, 0.0006. Similarly, for Mn in leaf, the highest BCF value was found in the Soneratia cacellaris. Overall, all the BCF value for Mn, zinc, and Pb in all 3 plant species were less than 1, indicating that these plants are not suitable for photo-extraction of heavy metal from the sediment. Photo-extraction is a process in which plants are used to remove and accumulate heavy metal from contaminated soil or sediment. Since the BCF value were less than 1, it suggests that the studied mangrove species do not efficiently accumulate this heavy metal from the sediment. Next, the study found that the F value varied between different plant parts. This means that Mn showed the highest translocation from root to leaf, followed by Pb and zinc. For Mn, the TF value were greater than 1 in all the studied plants except Nipah palm, indicating that these plants can actively take up terrace metal from the sediment and accumulate them in their aerial parts, making them potential candidates for photostabilization. Photostabilization is a process where plants help to immobilize or stabilize metal in soil, reducing their mobility and potential ecological impact. In conclusion, the average concentration of heavy metal in mangrove plant in order Mn, zinc to Pb, though all plant species studied had BCF less than 1, indicating a weak ability to accumulate heavy metal. For Tf, Avicennia alba and Soneratia exhibited value greater than 1 for Mn, indicating that these plants can translocate metal from root to leaf and act as phytoremediator in the study region. Lastly, the recommendation. First, need to use the instruments such as grinder to make sure the plant sample can be ground easily. Secondly, use variety of plant type to compare the ability of each plant to ensure more comprehensive understanding. The selection of the most effective plant species for specific contaminated environment. Thirdly, ICP-MS need to be used because it have more sensitivity, allowing for the detection of lower concentration of heavy metal in the plant sample. That's all from me. Thank you.